Well, welcome to day 27, students. Today we're going to be talking a little bit about DNA and kind of exploring the structure of DNA. And you already have been exposed a little bit to some of these ideas when we talked about macromolecules, but we're going to expand upon just this group of macromolecules. And so as we think about what DNA actually looks like, prior to the 1950s, people didn't really know specifically what it looked like, and biologists and chemists were working together, people like Maurice Wilkins and Rosalind Frank, to try and actually describe what DNA looked like. And they really kind of determined that DNA had these three primary pieces. And so I wanted to kind of identify what these constituent molecules actually were. And so the first one is this deoxyribose sugar. It's this kind of main kind of sugar that is central to this molecule, and you'll kind of see it here circled in red. In addition, they also have these phosphate groups. They always have this phosphate group. And then the third piece, of course, is this nitrogenous base. And so if we look at every single nucleotide, which of course we know is a monomer of DNA, they all have exactly the same deoxyribose sugar, they have the exact same phosphate group, but they have a couple variations on nitrogenous bases. They always have a nitrogenous base, but there are a couple different variations which we're going to explore. And so if we were to actually take a whole bunch of these nucleotides and print them out onto a piece of paper, maybe 20 of them, and cut them out and be able to kind of put them all together, we can kind of start to explore what an actual DNA molecule based on all of these nucleotides kind of monomers clumped together would actually look like. And so that's what you're going to see in just a moment, a video of me kind of trying to move these little paper nucleotides around. Now you're going to see in this image that I have here that there's these kind of thick lines in these shapes at the end of those. And of course those are not actually on a DNA nucleotide, but they're there to kind of let you know where they actually attach to other monomers of DNA. And the different shapes, whether they're kind of opened or closed, or they're these different types of shapes, these stars over here at this end, tells you that they need to attach to those same types of shapes on a molecule. And so you're going to go ahead and watch this video to kind of show how we might explore that idea.
Okay, so here you can see that we have these 20 nucleotides that were printed on paper and they were cut up. And so you can see that in this stack they are all identical, meaning the shapes, you can see they all have the triangles and the stars, and so the monomers are exactly identical in this particular stack. And so we're just going to take one of these, the top one, and save that. We can look at this other stack and see again that these are all exactly the same monomers. These ones in this case have these hearts in these squares, but the whole stack is the same. So we know that's going to be the same with these other stacks. So we're just going to take one representation of each one so that we can recognize we do indeed have just 4 different nucleotides out of this stack of 20 of them. And now the next step is to kind of match them together, and you can see those stars at the ends and then the shapes at the other end. And the key is that the stars have to match with other stars. The open have to match with closed, and the closed have to match with open, like you can see in this particular case. They always have to match one shape to one shape. They can't match to another. But we also have these other things at the end that don't match to anything else. And the goal is to make it such that all of these bonds have to match with something. You can see these don't match because they're not the same shape. And these don't match because they're also not the same shape. So we actually have to have these 2 nucleotides that come together, and they can match like this because there's an open and a closed triangle. And our goal is to kind of match all these things together so that our final product is one large molecule with these 20 monomers that are all attached to each other. But again, this doesn't work so well because we have those unattached ones off to the right. So we restored it again. We can see these all match together. There's an open and closed, open and closed. We can take these right here at the end, and those match up because we can have an open and closed, and those are all the stars. And we can clearly see those don't match, so we turn it around and the stars match up there. And so we can do this in many, many different ways, and you're going to see in a minute the couple different ways we could do it. Here's another way that we could maybe say, sure, those match there, the heart and the square. We have to kind of twist it around. Maybe instead we'll just have the stars all match together. So there's lots and lots of different variations.
And so now you kind of see how we can move these things around, and hopefully in the video you saw different couples of ways that we could actually put these things together. And following all the rules, there's a couple of different models or variations that would have actually been an adequate model. And so let's look at a couple of ones that students have produced in the past. So the first one is if we have 20 different nucleotides, then you can kind of attach them in this kind of nice linear fashion. And of course, since there's 20 of them, you can't really picture them all here, so that you can kind of envision they all go off to the left. Another model that a student made is to kind of compactly put them all here so that all those kind of different shapes are in the middle and all of the kind of stars are all on the outside of this molecule. Another potential one that I often see is the students will kind of put them together in a linear fashion, but they kind of hook them together in a loop so that there are no bonds left hanging on the outside. And so these are all kind of good and very different models of how we can take all of these monomers of DNA, these nucleotides, and put them together into one larger molecule. But before we kind of evaluate which one may be the best model, let's think about some of the assumptions that we made as we thought about some of these, putting all these things together. So hopefully the first thing you noticed was that if we look at this big pile of nucleotides, that there's only 4 different variations. As I already mentioned, they all share this exact same sugar and they share the same phosphate group. But there were 4 different variations of the nitrogenous base, as you can kind of see here. So we have these 2 different ones that have the triangles, and they have one triangle that actually has 2 rings, and this one only has 1 ring, and this one has 2 rings but it has the square in the heart, and this one only has 1 ring with the heart in the square. And so we can kind of think about the fact that there are these different variations or different types of nucleotides, but really there's 4 of them total. And the important thing about that is to know that there's a certain matching, that only one of these actually matches to another one, and this one can only match to this one. And so if we can kind of get rid of and strip away all those shapes and heavy lines that don't really exist, we can actually see how this pairing actually ends up looking. And so in this particular case, we've just focused on the nitrogenous bases. Clearly here's that base, and we know it attaches to the sugar that then also attaches to that phosphate, but we've just reduced it for simplicity here. And we've actually labeled each of those different types of bases. And so you can clearly see that there is this relationship that the adenines always are going to bond to the thymines, and the guanines are always going to bond to the cytosines. And there's this kind of nice pattern that they always bind together. And we know over here that this was where our squares and our hearts were, and these were our 3 different triangles that bonded together. And we know that this one has these 3 different places that it bonds, where these only bond in 2 different places. And in addition, another thing that's kind of different about the nucleotides you hopefully noticed is that these ones have these 2 rings, and these have these 2 rings, whereas this just has 1 ring, as does the cytosine over here. And each of those things has names. And so the nucleotides that have the 2 rings are what are called purines. And purines are kind of special because with those 2 rings, it makes them a little bit larger, so they kind of take a little bit more space, if you will, and kind of infringe upon the other one. And they match nicely, of course, with the ones that only have 1 ring, like the thymines and the cytosines, because they don't take up nearly as much space, so they kind of bond together. And this idea that these things are always going to be coming together is what we call Chargaff's rule of base pairing. That is, A's always match with T's, or T's always match with A's, and G's always match with C's. That's always how it works. And we can use this rule to actually answer and ask some really interesting questions. So if we kind of consider all of the DNA that's in a whole bunch of different organisms throughout the diversity of life, we can all understand that they all have A, T, C, and G. But maybe the distribution or the composition of those could actually vary. So as an example, if we look at humans, we can say that on average that we have about 30.3% of our DNA are adenines. Whereas we can actually compare that to a bacteria, E. coli, and see that approximately 26% of their DNA is adenines. So we can really see some differences between different types of organisms. But the thing that we can actually use Chargaff's rule for is to kind of make estimates on how much the other nucleotides comprise each of these DNA of these organisms. So if we look at humans and we say, well, 30.3% are adenines, then if we know that they always match with thymines, then we can make the assumption that, well, that means that 30.3% are thiamines, so that about 60.6% are A's and T's. Then that means that the remaining 100% have to be C's and G's. And so the math kind of works out simply by using Chargaff's rule, knowing that G always binds with C and T always binds with A. And so you can do this using E. coli to kind of make an estimate. If you know that 26% is adenine, what percent of the genome in this particular case is guanine? And so I suggest that you take some time to kind of do that calculation. If you finish it and you get 24%, fantastic, keep moving on. But if you don't, you might want to pause it and rewatch part of this video. So the other thing I wanted to mention is that the composition can vary, but keep in mind that this is simply the proportion of those. It doesn't mean that there's more adenines in human DNA, it just means that the proportion of adenines is a little bit greater. So now let's kind of go back to these models and think a little bit about what's the difference between all the different models as we take all those nucleotides and try to piece them together into one molecule of DNA. And we can, of course, go back to this idea that DNA is, the whole function of it is to store information. And so if our goal is to store information, as we think about each of these models, which one seems like it's doing a better job of storing information? And keep in mind that we can kind of think about the stats of the fact that we have lots and lots of DNA. If you stretch out all of the DNA in just one of our cells, it's a tremendous amount of it. If we just kind of stack it end to end, we know that within one single human cell, all of the DNA, if it's stacked end to end, would be about 6.5 feet long. So that's a lot of DNA. So the goal is to kind of compress that, all of that DNA, into this little teeny microscopic space in the nucleus. So as we kind of think about which one of these models does the best job, hopefully you have identified that this one to the right seems to compact, and you can actually see all 20 of those nucleotides in a very, very small space. And so that clearly is the best model. You can kind of see that we have these kind of two rungs on the side, and the nitrogenous bases kind of make these, the rungs actually that you step on the sides of the ladder are the sugar and phosphates. But another kind of thing to think about why this structure is so efficient is that it's not just that it's this ladder, but the ladder actually turns more into this kind of helical structure. So if we kind of take this model and say, well, this is what we started out with, this ladder where we have kind of these bases kind of connecting at the center. But they actually, if they start to twist, you can clearly see that they're even going to take up even less space. And so that's even a better thing about the structure of DNA, is that it does its kind of function, it is able to accomplish that very, very well by having this structure, this helical structure. And so as we think about how it is that it can form that double helix, there's a couple things that can contribute to making that a successful structure. And so first is looking at kind of where the bases and the sugars and the phosphates are actually located with respect to one another. And so first and foremost, all those bases that kind of form the rungs of the ladder, as we said, are hydrophobic, meaning they're kind of water-fearing, if you will. And so if they're kind of within this kind of curled up molecule in here, kind of right in near the center, then they're actually protected a little bit away from the outside, which is most of the cell is actually water. And so then the outside kind of sides of the ladder, if you will, are the sugar and phosphates, which are hydrophilic. So that's a really good way for them to kind of have this good structure. The other thing that contributes to this double helix is the asymmetry that's inherent in the kind of sub-pieces of this molecule. And so I already mentioned kind of the asymmetry in the purines and pyrimidines. And so we also kind of think in terms of if you're trying to stack all these nucleotides, what they end up doing is kind of displacing them a little bit to the side. And so having this helical structure allows for the bond angles that are necessary to actually attach all those monomers together. And so that kind of manifests is this structure that we know is the double helix, which is our DNA.